Oops, I accidentally hit the wrong button there. I didn't mean to stop the previous video. I wanted to show you the tortoise shell cat here from your book. So it's kind of a splotchiness of orange and black, and it's because of those two X chromosomes being randomly inactivated. And so I want you to write up here underneath this example, tortoise shell cat. So that's what they are referring to. All right, and so we are going to continue on with 15.4. And so 15.4 is dealing with having an abnormal number of chromosomes. And so far we've talked about how the traits are caused by, disorders are caused by the genes, the combinations that you have, but it also can be caused by the abnormal numbers of chromosomes. So large-scale chromosomal alterations, so it could be numbers of chromosomes or the structure. So chromosome number or having the right number, but something's wrong with the structure of it. So large-scale chromosomal alterations often lead to spontaneous abortions or miscarriages or cause a variety of developmental disorders. So sometimes it can be so drastic that a miscarriage happens or if the child is able to live, there may be some characteristics that allow them to have some problems, whatever the gene that is affected controls. So let's look at abnormal number of chromosomes first. In non-disjunction, pairs of homologous chromosomes do not separate normally during meiosis. As a result, one gamete receives two of the same type of chromosome and another gamete receives no copy. So this is a mistake. So let's look at how that can happen. So the mistake can occur in one of two different ways. So this is way number one, and this is way number two. All right, so here is meiosis one. The homologous chromosomes are supposed to separate. So this is a pair. One's supposed to go to this cell, one's supposed to go to this cell. Look at both of them go to the same cell. This one doesn't get any of chromosome number 2 here. And so therefore, if meiosis 2 happens properly, then if these two here have an extra chromosome, so that's why it says N plus 1. So they have an extra chromosome instead of having the right haploid number. They have an extra. And then this one here is N minus 1 because it's missing one of those larger chromosomes because of the non-disjunction or the not separating properly way back here in meiosis 1. So that is one way. A second way in order to get non-disjunction happening is if it happens in meiosis 2. So meiosis 1 occurs properly, so one small and one large chromosome goes into each cell. But instead here, if you notice here, both sister chromatids go to one cell instead of being separated into two cells. So now this cell has an extra chromosome. This cell is missing a chromosome. But if everything happened correctly here, this has the right number of chromosomes, which is N. So in humans, this would be a cell that would have 23 plus 1, so 24 chromosomes, and this would be missing one. All right, so those are mistakes that can happen that can cause abnormal numbers of chromosomes in their kid. Because let's say, for instance, if this is an egg and a sperm cell, everything happened correctly, so it's got one large and one small, then you're going to have a kid that has two smalls and 3 of those larger chromosomes. So you're going to have 3 of one kind of chromosome. And depending upon what chromosome you inherit 3 of, because one of your parents gave you an extra, determines what is the disorder or the disease that you have. So aneuploidy results from fertilization of gametes in which non-disjunction occurs. So we get an abnormal number of chromosomes. Offspring with this condition have an abnormal number of a particular chromosome. They call it a trisomic or trisomy trisomic zygote has 3 copies of a particular chromosome. So that would happen like I just went through here. A monosomic, mono means 1, tri means 3, zygote has only 1 copy of a particular chromosome. So if you only had 1 of chromosome number, 23 or something like that. And polyploidy, poly means many, is a condition in which an organism has more than 2 complete sets of chromosomes. So this is common in the plant kingdom. That means that they could have 3 of every kind of chromosome. Humans, if we had 3 of every kind of chromosome, the baby would not survive. So an example is bananas. Bananas are triploid. They are 3N. I'm going to put here only a few known cases in the animal kingdom. Certain fish and amphibians. Okay, so that's abnormal number of chromosomes. So I'm going to put here for trisomy example, trisomy 21. This is also known as Down syndrome. So this is when a person has 3 chromosome number 21s instead of 2. So therefore, non-disjunction occurred in one of the parents to pass that on. And the next thing is alteration of chromosome structure. Breakage of a chromosome can lead to 4 types of changes in chromosome structure. First one is deletion. Just like it sounds, removes a chromosomal segment. Duplication repeats it. Duplicates it. Inversion, when you're inverted, you flip, so it reverses a segment. And translocation moves a segment from one chromosome to another. So if we look over here, this picture shows you an example of that. So here's deletion. Example of a deletion. So what this is showing you is, in each of these cases, what the original chromosome looks like and after the change has occurred, so after the deletion. So what they did is just lettered the sections of the chromosomes so that we can see, remember each one of these is a piece of DNA, and so and they highlighted the part that's affected, so the darker part is what's affected. So if we look here, this section of DNA labeled D was deleted. And so therefore it's actually missing a whole section, so whatever genes are in there, it will be missing those genes. The duplication process, if we look here, B and C is highlighted, so A, B, C, D through H. Notice here, we have 2 B's and C's. So therefore, that piece of segment of DNA was duplicated, and you have 2 copies of those genes on there. All right, and so you have extra DNA there. And inversion reverses a segment within a chromosome. So this is the part that's affected here. And inversion is like if you take it out and you flip it. So it doesn't affect the A and the E through H, but if you notice here, the B, C, and D, it's reversed or inverted. And so therefore, this is a piece of DNA. This can be a problem because when you read the DNA to make proteins, you read it in one direction, and so therefore it reverses the direction, and so therefore it can be a problem in making the proteins that are supposed to be made. It's kind of like if you flip a sentence around while the letters are still there, if you still try to read it left to right, it's hard to decipher the meaning of it, and the same kind of thing with DNA. And then a translocation moves a segment. So it moves it from like one chromosome to another. So one to chromosome number 1 to chromosome number 15. So here, this is a whole other chromosome, so that's why the letters are different. All right, so each of them are made out of their own separate DNA that has different genes on them. And so if we look here, they have switched a piece. And so therefore, A and B and M, N, and O have translocated, or this is a reciprocal translocation, which means one piece goes to one cell, chromosome, and the other piece goes to the other cell. And so they've switched. That's why they're both doing it. A one-way translocation would be like the AB being attached to just the MN, like this stays intact and it attaches to it. All right, so let's look at some human disorders due to these chromosomal alterations. So alterations of chromosome number and structure are associated with some serious disorders. Some types of aneuploidy, that means abnormal number of chromosomes due to non-disjunction occurring in meiosis, appear to upset the genetic balance less than others, resulting in individuals surviving to birth and beyond. So some, it's deadly, some you can actually survive and yet have specific characteristics. These surviving individuals have a set of symptoms or a syndrome. A syndrome means that there's a bunch of symptoms that the individual can have, but they don't have to have every single one of them. They could have any combination of those set symptoms, and that's what's called a syndrome. So Down syndrome. Down syndrome is an aneuploid condition. So a mistake occurred in meiosis where one parent gave an extra number 21 that results from 3 copies of chromosome 21. It affects 1 out of every 700 children in the United States. The frequency of Down syndrome increases with the age of the mother, a correlation that has not been explained, although they find that the mother has these egg cells that sit in her ovaries for her entire lifetime since puberty, and they think that the egg ages, and that the older the egg gets, the more likelihood that a mistake, and therefore separating the chromosomes properly, becomes more likely. And so after the age of 35, they've noticed that the chances of a mother having a child with Down syndrome grows up dramatically. And then as you get to 40 and so on, the chances go up dramatically with the age of the mother. So I'm going to put here some characteristics of Down syndrome. So let's write that they're short, usually have heart defects that are correctable heart defects, developmental delays, so they don't reach milestones, and they have characteristic facial features, eyes, okay. And so let me show you from your book the young lady with Down syndrome here. And so this is her karyotype. If you look at, she would have 23 chromosome number 21s, so abnormal numbers of chromosomes. Then we can also have the aneuploidy of sex chromosomes. So non-disjunction of sex chromosomes produces also different conditions. So example is Klinefelter syndrome, the result of an extra chromosome in a male producing XXY individuals. And so for that 23rd pair, instead of having an X and a Y, which determines maleness, they have an XXY. So they have 3 pairs of chromosomes for that 23rd pair, but they are still considered male because the maleness is actually determined by the presence of a Y chromosome, not the absence, or not the an X and a Y, it's the presence of the Y. So having an extra X still makes this individual male because of the Y. I'm going to give you some characteristics. So small testes, they're sterile, which means that they cannot have kids. Some breast enlargement they can have. and may have subnormal intelligence. Another example, monosomy X, is called Turner syndrome. This produces, they call it XO females. That means that it's a female. Normal females, remember, have two X's. This female has just one X, and they don't have a second sex chromosome. So they put a 0 in there, meaning that they just don't have a second sex chromosome. They are sterile. It's the only known viable monosomy in humans. Viable meaning that they're able to stay alive and to live with that. Some other characteristics, their sex organs do not mature. Hence, up here, they're sterile. Doesn't affect their intelligence, normal intelligence. So this means that something happened either in mom or dad's meiosis process where they're missing a chromosome. So it's like one of these N-1 where the chromosomes didn't separate properly and so on. Another one, disorders caused by structurally altered chromosomes. One syndrome, Cry-du-Chat. This is a French words here. Cry, literally means cry of the cat, because the babies that are born with this are said to, their cry sounds like a cat cry. Results from a specific deletion in chromosome number 5. So they are just missing part of chromosome number 5, so they're missing part of their DNA, which then causes them to have the symptoms. A child born with this syndrome is mentally retarded and has cat-like cry. They usually die in infancy or very early childhood, all right, because of the genes that they're missing. Certain cancers, including certain types of leukemia, are caused by translocations. So this is a reciprocal translocation. Remember that this part here, so this is chromosome number 9, and let's do this part here. This is chromosome number 22. So they flip, it's reciprocal, they switch, so this part is now here on that chromosome, and this is now part of this. They call this the Philadelphia chromosome, and it causes the blood cancer called leukemia. So that's another example. Should put here, let's put a couple of things. This activates a gene that leads to uncontrolled cell cycle. And this is, like I said, it's a blood cancer. Let's put here that it occurs in mitosis of cells that will become white blood cells. All right, and before we go on to 15.5, I think this is where I'll stop for this video.